Good evening. Good evening. We are in previous class we discussing the cell organelles of the microscope, showing the homo special type of the cell cycle, cell apoptosis. Okay, next one. We are today we are discussing the genetic code. Today we are discussing the genetic code. What is the meaning of the genetic code? Okay. All genetic information is ended in DNA molecule and is later transferred into the mRNA. Okay. These are the few cases the DNA is the absent, but the information is continued genetic RNA. Genetic RNA. Most of the 20 proteins are the different kinds of the essential amino acid nucleic acid. They have only four different kinds of the bases. Already we discussed the purines and the pyrimidine. Okay, AGCT, Okay, actually is the uracil in replacement the thymine because the RNA. Okay, actually 20 proteins are the different kinds of 20 protein essential amino acid nucleic acid have the four different kinds of the bases. What are the four kinds of the bases? Adenine, cytosine, guanine, and next one uracil and thymine. These are the four bases have been considered as the alphabets of the language of nucleic acid, and 20 amino acids the alphabets of the language of the protein. Next one, the sequence of the nucleotides. The mRNA will decide the sequence of the amino acid and protein chain. And this is the what actually what is the meaning of the genetic code? The genetic code is the dictionary that is corresponding with the sequence of the nucleotide and sequence of the amino acid. Sequence of the nucleotides and sequence of the amino acid. The genetic code is the set of rules by which information encoded in genetic material. The genetic material DNA or RNA sequence. DNA and RNA sequences is translated into the proteins by the living cells. Actually, the genetic code, the term is given by the George Gamow. Okay. The genetic code is the term is given by the George Gamow. Okay, the genetic the term is given by the George Gamow. Already, what is the already we discussed the correspond with the sequence of the nucleotides and sequence of the amino acids is given by the George Gamow. And next one is the what is actually the DNA and the DNA is the they discovered by the Watson and Crick model. George Gamow is the postulate that the 3-liter code must be is the employed to the encode in the 20 strands of the amino acid. Actually, how many 21 amino acids is there? Okay, only 20 acids are the amino acids are there. Especially they can see in living cells of the build up proteins. Actually, genetic code is the letters is the whatever the base space already we discussed the A, G, C, T, and U. These are the corresponding the nucleotides found in the these are the only A, G, T, C are the corresponding the in A, G, C, T is especially the corresponding nucleotides found in the DNA. And next one A, G, and CU. These are the corresponding nucleotides found in the RNA. These are the organized into the codons. Okay, collection of the codons is also called as the genetics in the genetic codon. Collection of codons is also called as the genetic codon. Okay, genetic codon. Actually, how many amino acids there? 20 amino acids there. These are the how many actually 20 amino acids are the 64 codons. Okay, each codon should have the three nucleotides. Is the important specific specificity of the amino acid for the specific codon. Actually, one nucleotide is the 4 combination. Two nucleotide is the 16 combination. Three nucleotide is the 64 combination. Most of these are the set at the 20 amino acids. Actually, the 20 amino acids they are given to the 64 codons. Okay, 64 codons. 20 amino acids in the 64 codons. These are the genetic code. These are the special ATGC. Mainly is that the genetic code is they are found into the in DNA. Three bases are the code for the different types of the amino acids. Okay. Next one. These are the 64 codons. Actually, how many amino acids is there? 20 amino acids. These are the 20 amino acids are they are encoded by the 64 codons. How many stop codons? Three. UAA, UAG. Next one, UGA. Okay. What are the initiation codon? In methionine is the initiation codon. Okay. These are the initiation of the what are the initiation of the amino codon is the methionine is the AUG is the methionine, UAA, UAG, and UGA. These are the stop codons. Actually, for example, methionine only these are the indicate the only single codon AUG. For example, valine. Valine also there is the encoded by the 4 amino acids. The 4 codons is there. GUU, GUC, next one GUA, CUG. Okay, the especially the different types of the codons also. In the uh, uh, valine is then the fourth type of the codon. Only some methionine only there the only single codon AUG. Okay, next one is the different types of the codons, 64 codons. Okay, genetic code is the already we discussed the codons is also called the dictionary codons. Each codon consists of the 3 base pairs, the triplet codons. There are the 64 codons. Okay, 61 codons are the indicate the 20 amino acids in protein. 3 codons are do not code for the any amino acid. These are the stop codons. Okay, these are the stop codons. These are the they not indicate the any amino acids. Okay, these are the see this one is the UAA. These are the stop codons. These are the not indicate the any type of the amino acids. UAG also. These are the stop codon. Next one UGA also. These are the stop codon. They did not indicate the any amino acids. Okay, next one is the different types of the codons. See this one. 1 minute. Okay. These are the genetic codon. Actually, how many genetic codons is it? 64. The 61 is the indicated the 20 amino acids. The 3 is the indicated that there is no indicated any type of the amino acid. That the stop codon. Okay, stop codon. The next one is the next one. Four actually we discussed that the four base pairs are they have been the considered as the alphabets the language 20 amino acids. And next one, the genetic code works is the prepare a dictionary of the translating the language of the RNA and into the language of the protein. Okay, different types of the language of the protein. Next one, already we discussed the codon genetic code. What is the meaning of the genetic code? The sequence of the nitrogen bases in mRNA molecule, which is enclose the information for the mainly synthesis of the protein molecules. Synthesis of the protein molecules is also known as the genetic code. The next one is the what are the mRNA codes are for the particular amino acid is known as the codon genetic code. And next one is the genetic these are the codon especially they can give the particular amino acid. For example, AUG. AUG is the methionine. These are the indicate the only particular amino acids. Okay, one amino acid they are containing the different types of the codons. Okay, some are the stop codons also there. Next one, we'll see the genetic code was to the determine the exact number of nucleotides in the codon which code for one amino acids. Only some are the which code for the single code. For example, single code consisting only one nucleotide provides for the 20 amino acids. And next one, the two nitrogens double bond provides the 16. Index 3-1, especially George Gamow in 1984, the pointed out the possibility of the three letters. Possibility of the three letters is in 1954 is the genetic code. Mainly the genetic code, the term is given by the George Gamow in 1980, sorry, 1954. 1954. The George, these are the mainly how many amino acids are indicated? 20 amino acids. Those are the, for example, actually, he studied also, especially they can see in some of the pro children, they are content children. That's to stop on UAA, UAG. Next one, UAA, UGA, sorry, UGA. These are the all are the stop codon. These are the initiation codon. Methionine is the initiation codon. AUG is the initiation codon. Okay, next one. The triple codon is the most acceptable in most of the evidence, experimental evidence is the and the, both the genetic and the biochemical nature. The especially is the most acceptable codon is the triplet codon, triple codon. What are the meaning of the triple codon? AUG. These are the triple codon. Okay, AUG. Okay, UAA, UGC. Different types of. These are the mainly these are the triple codon. Next one, the genetic code is the experimentally described in the perfected indigenously by the Marshall Warren Nirenberg and the Robert Foley. Next one, Aravind Gopurana. Aravind Gopurana is the Indian scientist is the mainly jointly awarded in the Nobel Prize in Medicine in 1968 in physiology. Actually, is the synthetic RNA. These are the discovered in the Marshall Warren Nirenberg. Next one, Robert Foley is experimentally perfectly indigenous by the is got the Nobel Prize in 1960. These are the Aravind Gopurana is also these are the synthetic RNA. These are the discovered by the synthetic RNA. Okay, next one. What is the displacing of the genetic code? Displacing of the genetic code. The genetic code has been the cracked or distracted by the following kinds of the approaches. These are the three approaches of the displacing of the genetic code. One is the theoretical approach. Second one is the in vitro codon assignment. Next one is the, the in vivo codon assignment. In vitro codon assignment. And next one is the in vivo codon assignment. What is the meaning of the theoretical approach? The theoretical approach is mainly is the given by the George Gamow. Okay, George Gamow mainly proposed by the 1954 a triangle codon suggested by the exhausting of the theoretical framework in the different aspect of the genetic code. Theoretical approach also the mainly the proved by the mainly the George Gamow. The next one is the Gamow is what are the properties of the genetic code? What are the properties? These are very important in UGC and set net examination point of view. Triple codon is the corresponding to one amino acid of the polypeptide chain. Only triple codon is corresponding to the one amino acid of the polypeptide chain. Next one, direct template the translation by codon amino acid in the pairing. Direct template translation by codon amino acid pairing and translation of the codon in overlapping manner. These are the all properties, all characteristics of the genetic codon. These are the very important. Triple codon corresponding the only to one amino acid of the polypeptide chain. One amino acid of the polypeptide chain. Next one, direct template translation by codon amino acid pairing. Translation of the codon in overlapping manner. The overlapping manner is very important. And next one, degeneracy and overlapping. What is the meaning of the degeneracy? The amino acid being coded by the more than one codon. More than one codon. Already we discussed the in previous table. See, these are the degeneracy. For example, AUE, AUG, AUA. These are the indicated for the leucine. Okay, leucine. leucine also there the one amino acid and they can indicate the in polypeptide one nucleotide. For example, the degeneracy means that the for example a GUU, GUC, GUA, GA that they indicate the one amino acid. There are four triplet codons. Okay, that the degeneracy, overlapping, and degeneracy, overlapping and degeneracy. Only one amino is an amino acid being coded by the more than one codon. Next one, polyinteratory. Polyinteratory is the nucleic acid and the primary protein synthesized. Next one is the universality. These are the very important. Universality of the code. What is the meaning of the universality? The code being essentially the same for different organisms. The code being the essentially the same for different organisms. You know, these are the characteristics of the genetic code. These are the all properties. What are the properties? Triplet codon, direct template translation, overlapping, degeneracy, polyinteratory, and next one is the universality. These are the four properties of the according to theoretical approach. According to George Gamow, the codon is the character genetic code. These are the properties of the genetic code. The next one, in vitro approach. The in vitro codon assignment. This is the three parts of the in vitro codon assignment. One is the discovery and use of the polynucleated phosphorylase enzyme. Mainly discovery and use of the polynucleated phosphorylase enzyme. Okay, discovery and use of the polynucleated phosphorylase enzyme. And next second one is the codon assignment with the unknown sequence. These are the codon assignment to the unknown sequence. Assignment is the homopolymer and heteropolymer. Homo means same, hetero means different. These are the codon are the assignment of the homopolymer. Codon assignment by the heteropolymer. Next one, assignment codons with the known sequence. Known sequence, especially the known sequence also. These are the three parts are available in the in vitro code assignment. Next one, we discuss the each and everything. Discovery and use of the polynuclear phosphorylase enzyme. These are the mainly mango in the Silvio Okawa is isolated enzyme from the bacteria, Azotobacter vinilidae and the Micrococcus lepidococcus. These are the catalyze the breakdown of the RNA in bacterial cells. Mainly these are the phosphorylase enzyme, polynuclear phosphorylase enzyme. Mainly these are the catalyze the breakdown of the DNA, RNA bacterial cell. The enzyme is also called as the polynucleated phosphorylase enzyme. Okay. Both the scientists, the Gorenberg in the mango and next one Silvio Okawa is isolated enzyme from the bacteria. What type of the bacteria? Azotobacter vinilidae and next one is the Micrococcus lepidokinetics. Both the bacteria isolated from the mainly polynucleated phosphorylase enzyme. Next one is the mainly is the high concentration of the ribonucleotides. The reaction could be driven the reverse and the RNA could be the mid of the Botano in 1989. Okay, these are the codon assignment. Mainly discover the use of the polyphosphorylase enzyme. Next one is the codon assignment is the unknown sequence. Known sequence and unknown sequence is the assignment by the homopolymer. The first two codon assignment was proved by the Nirenberg and the Heinrich Metheve in 1961. Mainly these are using the in vitro system of the synthesis of polypeptide using the artificial synthesized mRNA. Polypeptide, artificial synthesis mRNA, especially the homopolymer, only one type of the nucleotide. Homo means same, only one type of the polymer synthesized. The codon assignment by the homopolymer. These are all the first clue codon assignment was proved by the Marshall Nirenberg and the Heinrich Methave. Mainly these are the used in the in vitro system of the synthesis of polypeptide. What are these are the using to the artificial synthesized mRNA molecule contain only one type of the nucleotide, also called as homopolymer. Okay, next one, the experiment repeated using the synthetic polynucleotide polymer chain, the codon assignment for the homopolymer, and next one codon assignment by the heteropolymer. So these are the next one is the genetic code to place the using the synthetic messenger RNA containing the two kinds of the bases. The technique using the laboratories Okawa and the Nirenberg, and the lead to the reduction of the composition of the codons for the 20 amino acids. The synthetic messengers are the contain the bases at random, also called as the random copolymers. Okay. Next one, random copolymers using the U and A nucleotides. Eight triplets is using the positive such as the UUD. Mainly, it's the random copolymer using the only U and A. Only there the two base pairs are they can use in the random polymers, copolymers. U and A nucleotides. Eight triplets are the positive such as the what are the UUA, UUA, UAA, UAU, triple A, double AU, A triple W, and the AUA. There are the how many? Eight codons. See this one, eight codons. Mainly is that the indicate the only U and A nucleotides. Eight amino acids are the for example, these are the only six eight amino acids. These are the eight codons. Actually, these are the only six phenylpheniline, leucine, tyrosine, lysine, and asparagine and isoleucine are the codons. Next one is the assignment is the known sequence. Already assignment with the assignment codons of the unknown sequence, homopolymer and heteropolymer. Homo mean only single RNA. Only single RNA. They can form single RNA. Next one is the heteropolymer. Different types of the heteropolymer also that indicate only AU codons. Okay, heteropolymers. Okay. Next one, the assignment of codons with the known sequence. Known sequences are the mainly are the trinucleotides. They are using of trinucleotide are mini messengers in the filter binding using of the trinucleotides. Next one, mini messengers in filter bonding. What is the filter bonding? The ribosome bonding technique. Ribosome bonding technique. Ribosome bonding. There are also techniques are developed by the Nirenberg and Leder. Mainly they are using the finding of the amino acid tRNA molecules, especially bind with the ribosome RNA complex. Next one is the binding does not require the presence of the long mRNA molecules. The association of trinucleotide mini messenger with the ribosome of sufficient cause of the amino acid tRNA binding. Mainly these are the assignment codons with the known sequence, unknown sequence, and known sequence. Known sequence, especially use of the trinucleotides, mini messengers in filter binding. What are the bindings? The ribosome binding technique. Okay, ribosome. Next one is the already in the theoretical approaches. Three types of the approaches there. Theoretical approaches, and next one the in vitro codon assignment and in vivo codon assignment. These are the final the three kinds of the technique is the different molecular biologists that determine same codon is in the in vivo amino acid replacement studies of the mainly main are the amino acid replacement. There are the three kinds of the techniques used in the in vivo codon assignment, amino acid replacement. Next one, frame shift mutations, frame shift mutations. Next one is the comparison of DNA or mRNA nucleotide cryptogram with its corresponding polypeptide. There are three kinds of the techniques in the in vivo codon assignment. In vivo codon assignment. What are the properties of the genetic code? Already we discussed these are the characters are the very important. Genetic code is the following the properties. What are the properties? The code is the triplet. Already we discussed the AUG, UAA, UAG. Different as a okay. Some are the mainly the characters. Are the characteristics of the genetic code. Code is the triplet. Code is degenerate. Code is no overlapping. Okay. The code is the commaless. The commaless. The code is non-ambiguous. The code is the universal. The code is the polarity. Code has act the start code. The code has act as a stop code. Start code. AUG is the stop code. Next one UA is the some of the stop code on. Okay, these are the properties of genetic code. These are the characteristics of the genetic code. Triplet, degenerate, non-overlapping, commaless, non-ambiguous, universal, polarity, act as a start code AUG, act as a stop code UAA. These are the properties of genetic code. Triplet codon the term is given by the Gamow 1954. The letter was first by the Gamow. The codon presents a genetic code that specifies one amino acid the polypeptide chain comprised the sequence 3 nitrogen bases on mRNA in a specific sequence. The first experimental supporting the concept of the triplet code was proved by the Crick and the co-workers in 1961. T4 bacteriophage. The triplet code are mainly experimental evidence. The concept triplet code is proved by in T4 bacteriophage. Degenerate. In particular, amino acid is coded by the more than one codon. It's also called as the degenerate. Already only AUG is the only they contain the only single codon AUG methionine. Okay, the degenerate mean the particular amino acid is coded by the more than one codon. More than one codon. For example, except the tryptophan and methionine, these are the only they contain the only single codon methionine AUG. Okay, tryptophan and the methionine only they contain the only single codon. The next one is the single codon. All other the 18 amino acids are the more than one codon. How many amino acids? 20 amino acids. The methionine and tryptophan. So what are the important? Tryptophan is the precursor of mainly oxins. Next one, tryptophan is the mainly precursor of the oxin. The methionine also is the precursor of the some of the protein synthesis. Okay, methionine, the tryptophan, the actually only single codon there contains tryptophan and methionine. Other than 18 amino acids, except the two codons only they contain the single codon and the 18 amino acids they contain the more than one codon. More than one codon. The degenerate. The codon degeneracy is two types: partial degeneracy and next one complete degeneracy. Partial mean it is the nucleated identical, but the third nucleated is the degenerate codon refers the CEU and TUC is the codon used for the leucine. Okay, partially degeneracy, complete degeneracy. The complete degeneracy is the third position and the still code the for same amino acid. See, this one is the U. The next one is the code for the assignment of serine. Okay, complete and partial degeneracy. Okay, next one. Multiple the different amino acid is given to the some degeneracy genetic code. Origin and use origin and use in the serine, the contains each six amino acids. Okay, the total codons is the 18. Next, alanine, glycine, proline, threonine, valine. These are the four number of codons of each amino acid is the four. Next one, how many total number of codons? 20 amino, the 20 codon. The next one, isoleucine, the stop codon, the 3. Isoleucine, the stop codon, the number of codons, 3 codons. Okay. Aspargine, aspartic acid, lysine, phenylalanine, next one, the tyrosine. The indicate each two number of codons for each amino acid, the 2, 18 total number of codons. Next one, methionine, the tyrosine. These are the two. Only these are the indicate the single codon, methionine and next one tyrosine. Sorry, tryptophan. Okay, next one is the codon non-overlapping. For example, that means that the same letter is not used for different codons. Same letter used in the different codons. For example, cat. Okay, cat and next one TGA, next one ATC. ACA, next one TGA. Okay, the non-overlapping. Overlapping codon. These are the overlapping codon. CAT, then next one, then start with the TGA. CAT, next one TGA, next one is the ATC. These are the overlapping codons. These are the non-overlapping and overlapping codon. These are the overlapping. The codon of non-overlapping, which means the same letter is not used for different codons. Different codons are the properties of the genetic code, non-overlapping. Genetic code is the mRNA begins at the correct point and there is no overlapping of codons. Genetic code is the non-overlapping. These are the non-overlapping, but the bacterial phase 5174. There is no possibility of the overlapping genes and the codon. Next one is the code is the commaless. Okay, these are the property of the one of the property of the genetic code. There is no punctuations or commas between the two codons in the genetic code. The codon is the reserved for the function. Okay, there is no commas. The codon, no commas. The punctuations and the commas, there is no between the two codons. That the property of the genetic code. Next one, the code is the non-ambiguous. Okay, the genetic code is the inside of the cell medium in vivo. In vivo said to be the non-ambiguous because the particular codon always code for the same amino acid. Particular codon is the code for the same amino acid. More than maybe the code for the more than one codon, more than one codon is the degeneracy. Okay, the codon is the non-ambiguous. Next one is the code is universal. The one type for the mainly genetic code is the found valid for the all organisms ranging from the bacteria to man, such as the universality. The code is that by the Marshall and the Kasky Nirenberg. Mainly are the found in the E. coli bacterium. The Guinea pig mumble amino acid. Acetyl tRNA is the almost of the same code. Acetyl tRNA. The code is universal. Okay, universal. These are the properties of the genetic code. Next one is the code has the polarity. For example, the code always read the fixed direction 5' to 3' direction. Okay. The codon is the polarity 5 to 5. The mainly is the from left to right and the right to left with the specific different amino acid. Okay. The code is the polarity 5' to 3' to 3'. Next one, the some codes act as the start codons. What are the starting codons? The most of the organisms, most of the prokaryotes and the eukaryotes also, AUG is the starting codon. Okay, it's the starting AUG. Methionine, the starting codon, initiation codon, polypeptide change is the methionine and the formal methionine. Next one, GEG code for the value, but when the normally AUG code is the last for the deletion only. U is the initiation codon. There is no availability of the AUG, then put the GUG is the initiation codon. Next one is the stop codons. These are the three types of stop codons: UAG, Umber, Oaktree, Opel. Umber, Oaktree, Opel. The three are the termination codons, also called as the termination codon. The do not code for the any of the amino acids. Okay, these are the stop codons. UAG, UAA, UGA, Umber, Oaktree, Opel. These are the name of the stop codons. Already we discussed the initiation codon is the AUG. The next one is the termination codon. The initiation is the mainly are the mechanism for the protein synthesis. The initiator and the terminator codons known as the signaling. The phenomena is known as the punctuations. Punctuation. The punctuation helps the delimiting and the different cistron of the polycistronic mRNA. The signals at the spaces do not occur between the codons of the message, but the initiate and the technology section. The message between the functional gene, initiation of and termination of codon. Signal codons are the codons that the code for the signaling during the protein synthesis is also called as signal codon. The mainly are the codons the mainly signaling during the protein synthesis is known as the signal codons. What are the signal codons? AUG, UAA, UAG, UGA. Okay, these are the signal codons. Signal codons and the next one stop codon. Initiator codon, initiation codon is AUG. Next one is the four are the signal codon. Okay, UAA, UAG. Next one is the okay, these are the signal codons. Mainly these are the codons during the protein synthesis is known as signal codons. What are the three? These are the four codons are the signal codon AUG and the some are the AUG. Next one UAA, UAG, UGA. Okay. Stop codons and next one is the initiation codons also total is the all are the signal codons. The signal codons are the two types: initiation and the termination. Initiation means starting codon. Termination means stop codon. Already we discussed the initiation codon AUG, stop codon the UAG, UAA. These are the signal codon is the AUG. Sorry, initiation codon. Mainly these are the starts the translation process known as the start codon. It also known as the initiation codon. Initiation codon. Next one mainly the synthesis of the polypeptide chain. Synthesis of the polypeptide chain. Okay. Already initiation codons, four types. Initiation codon is only one. AUG is the initiation codon. Is the first codon messenger RNA translated by ribosomes. Already the protein synthesis related to the ribosome. Protein synthesis. The initiation codon is the AUG and its code for the amino acid methionine in eukaryotes, the formal methionine in prokaryotes. In 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 eukaryotes, especially the initiation codon is the AUG. AUG is the eukaryotes in the methionine. In prokaryotes is the initiation codon is the formal methionine AUG. The two only AUG is the initiation codon. In eukaryotes and the prokaryotes is the different. In eukaryotes is the methionine. In prokaryotes also called as the formal methionine. These are the initiation codon. Next one, some cases are the instead of the AUG, there is no availability of the AUG, the GUG. Okay, G is the valent. Valent is the also part of the signaling, signal codon, GUG. G is the initiator codon only about the 13th frequency. AUG initiated in the E. coli bacteria. Next one is the okay. These are the initiation codon. Next one, termination codon. What is the termination? Is that the provide signal for the termination of polypeptide chain. Is also known as the termination codon and the stop codon. Already discussed the stop codons and termination codon. There are the three types of the termination codon. UAA. UAA is also called as the OCRI. UAG is the AMBER. UAG, the AMBER. Next one, UGA is known as the OPEL. OCRI, AMBER, OPEL. The three are the stop codons or the termination codon. UAA, UAG, UGA. The different types of the names. UA is also OCRI. UAG known as the AMBER. UGA also is the OPEL. Set examination point of view, then ask from the UAA and the UGA and next one UAG. Next one, stop codons also not code for any amino acid. There is no earlier code. The nonsense codons is also called nonsense codon. UAA, UAG, UGA is that the nonsense codon. What are the initiation codon? Is the AUG. Nonsense codons are the UAA, UAG, UGA. Okay. Next one is the signals of the stop or termination codons are related to the by protein called the releasing factors. These are the release factors RF1, RF2, RF3. Most of these stop signals are not read by the tRNA molecules. Stop signals are not read by the tRNA molecules. Prokaryotes, they release factors are the RF1, RF2, and RF3. And especially in prokaryotes, the stop codon, the releasing factor signal stop or termination codons are the protein. By proteins is also called release factor. There are three types of the release factor in prokaryotes: RF1, RF2, and RF3. Okay, next the RF1 is recognized the stop codon UAA and UAG. They are the stop codon recognized. Mainly RF1 recognize the UAA and UAG. Next one, RF2 recognize the UAA, UGA. RF1 also they can recognize the UAA. RF2 also they can recognize the UAA. RF2 mainly RF2 recognize the UAA, UGA. Next one, the RF3 is the stimulate the RF1 and RF2. Actually, RF1 they can indicate, they can recognize the UAA, UAG. RF2 recognize the UAA and UAG. That the both are the stimulated by the RF3, RF3 releasing factor. Especially there are the three factors are only the carriers. Okay, in the prokaryote protein, this protein RF1, RF2, RF3. Okay, in eukaryotes also signal release factor RF recognize all the three stops codon. Eukaryotes also they can recognize the release RF factor, releasing factors. Next one is the next one's transcription. In the synthesis of RNA is the complementary strand of DNA is known as the transcription. Actually, RNA and DNA and protein transcription and the translation. Transcription and translation. Transcription means RNA they can convert the DNA is known as the transcription. Transcription takes place the three steps: initiation, next one elongation, next one termination. There are three steps are involved in the transcription process: initiation, elongation, and termination. What is the initiation mean? There are the three types of the mainly there are three different RNA polymerase in eukaryotes and transcription. Transcription mainly the DNA helix temporarily uncoiled by the synthesis of the RNA. Synthesis of the RNA mainly is the catalyzed by the enzyme RNA polymerase. Initiation, elongation, then next transcription. Next one is the translation mean. Translation means the genetic information of the into specific RNA molecule which is translated into specific protein. Next, mRNA, they can convert the DNA is also called as the transcription. DNA, they can convert the proteins. Okay, mainly proteins also called as translation, transcription, and the translation. Okay, mainly these are the translation also translate into specific proteins tRNA is known as the translation. Translation is the three types of the RNAs is available. One is the ribosomal RNA. Next one is the transfer RNA, the messenger RNA, mRNA. These are the made into the DNA templates. And next one, RNA polymerase also catalyze the synthesis of the RNA. Uh, Already four types of the base pairs are available to the DNA. AGCT, uracil is the replacement of the thymine. You can see the RNA U, A, and G and C. Okay. In DNA, there contain the AGCT. Okay. In the teeth replacement, they can use the uracil in the they can see in the RNA. RNA is three types of the RNA. Next one, mRNA. What are the importance of the mRNA? These are the single-strand mRNA molecules from the small amount of the approximately 5% that contain the mRNA. 100% of the RNA, only the mRNA amount is the 5%. Okay, the heterogeneous molecule size they contain the approximately 900 to 1500 nucleotides are in mRNA. Next one is the this is the carries the genetic message to the ribosome. Ribosome, the name is the messenger RNA. RNA synthesized rapidly, broke down the rapidly. A single mRNA strand is the ability, the function of the several ribosomes in the simultaneously. The presence of the small amount of the cell. Many ribosomes of the attached to the strand mRNA is also called as the polyribosome. Polyribosome. Next one is the rRNA, is also called as the ribosomal RNA. Polyribosome. Next one is the rRNA. rRNA is the ribosomes are the site of the protein synthesis, mainly rRNA, ribosomal RNA. Ribosomes also mainly observed in the Robinson the Brown in 1953 plant cell. Actually, is reported by the Palade in animal cell. Ribosome. Ribosomes are the mainly after the pre-cytoplasm is also attached to the endoplasmic reticulum, rough ER and rough ER especially R RER. Rough ER mainly the protein synthesis. Okay, protein synthesis, endoplasmic protein synthesis. 80 types of the ribosome in the eukaryotes, in the 70s ribosome in the prokaryotes, two subunits. 70, for example, 70s ribosome they contain the two subunits in the 50s plus 30s the 10 per units. Okay, that one. Next one, tRNA. tRNA is also called as the soluble RNA. Next, sorry, rRNA is also known as the soluble RNA. Is also called as sRNA. Okay, sRNA. Approximately the total RNA is 15%. tRNA they contain the 75 to 95 nucleic bases that contain the tRNA, transfer RNA. Actually, the complete nucleic sequence, tRNA structure of the tRNA is the clover structure. Actually, how many loops are present? The four types of the one is the initiation, the acceptor stem, and the APC loop, and the next one the variable loop and anticodon loop, and the dihydrocell loop. The dihydrocell loop is the small group of the arm. Okay, small group of the, especially in acceptor stem, especially they contain the first one alanine, alanine, the pi prime and the 3 prime is available. 3 prime they can attach the some of the genetic codon, some of the codon. Okay, the genetic code also. The next one, genetic code and the mechanism of protein synthesis is over. Next one, Mendelian inheritance. Next chapter is the unit 8, the Mendelian inheritance. Mainly 1900, especially George Mendel is the discovered the Mendelian inheritance. The some of the co-workers Mendel discovered by the independently rediscovered the three by Igor Drevis and the Carol Corans. And next one is the Tesmark is Austria. Is the three scientists, especially the Mendelian inheritance, the rediscovered by the some of the three scientists. Next one, Mendel experiments. Mainly is the Mendel experiments are the choose for the purulence of the garden piece and the 7 pairs of the contrasting traits used for the these experiments. For example, the all offsprings of the first filial generation F1 is the tall. It indicates the character is the tallness of the dominant of all darknesses. For example, the phenotypic ratio of the F1, mainly in the F1 generation is the monohybrid phenotype is 3:1, and the genotypic is the monohybrid, especially 1:2:1. Okay. Mendel proposed also law of segregation. Law of segregation. Segregation means separation. These are the pair of the alleles and the brought together the hybrids. There the mainly are the separate of the each of the hybrid forms gametes. Separation of the hybrid. Separation. Segregation means separation. Next one is the Mendel segregation is the fundamental principle of heredity and also termed as the law of purity gametes. Law of purity gametes, heredity and law of purity gametes. Monohybrid crosses. Monohybrid crosses the phenotype ratio 3:1 and the dihybrid genotype ratio the 1:2:1. The dihybrid crosses the phenotypic ratio is the 9 is to 3 is to 3 is to 1. Okay. Next one is the genotypic ratio. Hybrid class genotype is the 1 is to 4 is to 4 is to 2 is to 1, 2 is to 1, and 2 1. These are the genotype ratio of the hybrid class. Next one is the test class. What is the meaning of the test class? Test class is mainly determine the homozygosity or the heterozygosity, the dominant phenotype. Okay, the test class is also individual with having the recessive phenotype. The two pairs of the mainly they can cross with the recessive in F1 generation. They can, for example, capital T, small t. Okay, these are the capital T and capital T, small t is the cross with the recessive characters. Recessive character is also called the test class. Recessive characters. Text class and the back class. Back class also the mainly recessive characters and this is the dominant. Okay, recessive characters is the test class. Test class in the next one, phenotype. One minute. Mendelian genetics, Mendelian inheritance. Figure John Mendel, and these are the Austrian scientists. On Next one is the modification of the 3:1 phenotype creates the incomplete dominancy, codominancy, and the multiple less. Mendel experiment, Mendel this. Mendel started the physical traits of the inherited as the particles. Mendel did not particles in the chromosome or DNA. Genetic trait means the characters that can be passed from the parent offspring. Heredity, the passing of traits from the parent to offspring. Okay. Monohybrid cross is the involving the only single trait, and a dihybrid cross is the main involving the two traits. Different types of the two color, shape of the mainly they are using the Punnett square, solving the problem. Punnett square, Punnett box. What is the meaning of the alleles? The two forms are allele, dominant and recessive. Especially in the test cross mean in F1 generation, the can fuse with the mainly test cross only recessive alleles. This is the test cross, and the cross with the dominant character is also called as the